You already introduced me. The first,
Yeah, hello.
yes, I can start. I can start right away. Should I introduce myself again, or should I just start talking and let everyone see what's going on? Because there was a demand from, not really a demand, but I guess request from the participants about the applications and how bioinformatics actually make itself into the thing that we can help either save lives or help promote certain things in agriculture or whatsoever. So I decided to actually bring some of the work that I've done over the past couple of years here to share with you, and perhaps you will see something great about what we did, not myself, because I have to credit to the people who's behind the work that I'm about to show to you. It's still an ongoing process. It's not really done yet. It's going to last about like 2 or 3 more years, right, before everything ends. So I'm just talking a bit so that waiting for people to actually come in and sit down and get into the mode of listening to the talk. So you probably have met me the first day when you arrived here. And I'm thankful for the people who's behind this, making this possible for you guys to come to Thailand. And we can also show and share our hospitality to all of you. And there are still people coming. So anyway, it's my bad because I didn't tell Amit and Travis that I'm here on time, but I was just showing up to the team behind. He just came in and checked if I'm already here, and he found that I'm here. So, but anyway, there will be 2 projects that I'm going to share with you. I'm not going to go into detail on the theory behind the theory behind that make that happen. But the first project that I'm going to discuss about is going to be on the medical genomics. Some of you who are actually from the States, you will probably know this kind of project in the names of all of us, which is the Precision Medicine project that President Barack Obama announced in the year 2015, right after the David Cameron announced that he would initiate the Genomics England project. So the project like this started like a while ago, but it doesn't actually get into every country because it costs a lot. You know, the time they announced the project back in 2015 or 2014, the cost of sequencing is still high. You know, consider that, you know, like the countries around this area, you know, Thailand, Indonesia, Malaysia, even in ASEAN, we don't think that that will be possible. But after the year, I think 2018, I think we start seeing the price drop dramatically of the whole genome sequencing. And you have actually a chance to visit some of our facilities. You see that there are some sequencers, you know, in NASDA, and those sequencers are used to be very expensive and providing a very expensive service. Okay, so we got a chance to do this project. The project, okay, started since 2019, but at a time we, you know, had to go through red tapes and also a certain protocols that we need to do. Like, for example, finding someone who would provide us with the sequencing service. That is easier said than done. We cannot just go to the market and say that, okay, Illumina, you're going to be my guy. You will perform sequencing for us, or I'll go to Thermo Fisher and ask Thermo Fisher to offer that to me. Because, you know, when it comes to a national project, there are certain protocols that we have to go through in order to select the right company that will provide us with the service. So usually it's the price. If the price is right, then that would be the case. So in this project, the project estimated at the beginning is 4.47 billion baht. So you divide it by 35, you will see how many millions US dollars of the project that would be. So it's actually allocated to me as I'm a PI, approximately 110 million US dollars. So that's for myself. So that would include all those HPC that you probably get to see yesterday. It's not yet completed because we procure it timely. We're not going to buy everything at once, okay? So that is coming. So we are going to sequence 50,000 individuals. And the estimate depth, I'm not sure, you already talked about the genomics, right? It's about 30, but it can go up to 40, depending on the quality, because I'm also the one who set the bar very high. So even though that on average is 30x, but when I set the quality criteria, it will go beyond 30x for sure. If you know, like Q30, Q20, that's the quality of the base call. We also set another bar, which is the coverage of the Q20 and Q30. So the entire genome, the Q20 or Q30 has to be at least 80% or 85% coverage. All those kind of things actually set the bar to be so high. And we got a very good price. If you look at the price here, let's say how many US dollars divided by 30, maybe 300 something USD, yeah, or even less. I guess this is cheapest in the world at this point, even cheaper than Nebula Genomics from George Church that subsidized a lot. But I guess, you know, they dropped shirt and pants just to get this project. Because once they get this project, somehow they can land the account in this region and hope to be able to be the main service provider for this. Okay, so we're going to be using the DNA nanoball technology from Complete Genomics, but somehow BGI took over, I don't know, 5 years ago, just to get their brand on it. And right now they actually advanced this machine so that it will, you know, provide the read length even longer than 100 base pair. It used to be just only 100 base pair. Now it's 150. So we set this up. Okay, the file size is much larger than 100G. Due to the quality criteria, the file size is like 20% or 40% more than what I estimated. So on the computational side, it has to be adjusted as well. So that is large, it's huge, okay? We're focusing on the 5 disease groups. So basically everything. If you're not hit by the car, then it's covered. Starting from what? Rare group, rare and undiagnosed disease. So all the anomalies that happen when you are in early onset, everything will be included, right? And if you look at this one, this group, even though they call rare disease, but it's the group of diseases, right? Group of diseases. So there are many diseases, and we haven't actually found them all yet. It's very hard to find. So that's why we call them rare disease. But if you put them all together, it constitutes about 8% of the population. This is a lot. It's not less, okay? It's a lot. The second group, of course, the cancer. That, you know, is the second leading cause of death, right? Nowadays, and surprisingly, you know, like breast cancer, we thought that we are not going to be so much of the patient with breast cancer, but since this project started, we found that a lot of the Thai individuals have either the mutation, like BRCA1/2 mutation, like Angelina Jolie. It's quite common. among the people in Thailand as well. And that means perhaps people around this also have the same problem, but it's just that it's under the carpet. We never actually lifted it up. We never actually checked it. So we don't know if that's going to be. Another group is the pharmacogenomics, and that's so common among us. Some of the drugs that we have taken cause us to have the adverse event, not just only something that you have adjusted dose, something you don't even know why it happened. We call idiosyncratic adverse drug reaction. The Stevens-Johnson syndrome. You take a drug like carbamazepine, which is the epilepsy type of the drug suppressing the seizures. If you don't check your HLA first, this HLA that you have to check is the HLA B 5201 thing. That's the thing you have to check. Even the allopurinol, which is the medicine for the people like my age taking, gout. I'm not having gout, by the way, but most men who's 50 and above usually develop whatever disease called gout, and that you have to take the allopurinol. And if you don't check your gene, then of course you also develop the adverse drug event as well. The NCD, the non-communicable diseases, that's diabetes, hypertension, coronary artery diseases, many things. And the last one is infectious, where we're talking about the host factors, like when you get COVID, some people develop a severe COVID, some people develop mild COVID, and why would that happen? So everything is all in your gene, right? So that's me standing, but the cartoon. But this actually gives you an idea. If you study genes, what will you get from it? If you look at these triangles, the two triangles forming a rectangle, you will see that the spectrum on your left-hand side, the red one, is a genetic, right? A lot of genetics, all the way to little genetic or non-genetics. The blue one is an environment. So from no environment effect all the way to a lot of environment. So on this end would be I have a cut. I just cut myself with a knife. Of course, I'm sane. I'm not insane. I just accidentally cut myself. So that would actually be pure environment. Or just walk and a rock just dropped onto my head, and that's pure environment. On this end is pure genetics. So you're born with a genetic anomaly. Okay, you just congenital defect, like having loss of hearing at birth. Okay, or having some malformation in your mouth, or having extra fingers. All those pure genetics. It happened when it's early onset. This happened when it's late onset. So what are those diseases that happen in the late onset? Diabetes, hypertension, right? Many other, even including cancer. So the genetic factor play roles in the entire spectrum, starting from pure genetic all the way to not pure environment, but to that end. So if we can actually identify, I call it culprits. Okay, if you come with a birth defect and you identify the culprit at birth, some of them are preventable. So you're not cured, but you are, but it's preventable. Like some of the defect, neurological defect, you can you can be treated by giving some form of the vitamin supplements that will actually get you to be healthy again. But you have to actually, of course, taking that kind of thing. But you have to know that at birth. If you do not know that and let it be, then of course something will happen to your brain. I've got, I'm not sure if in the slide, but I just talk about it. I met this girl, she's a baby, five months, in the year 2000. she's born and she's like having acidosis. She's like having, tube all over her bodies, and we don't know what happened to her. So my friend, Professor Warasak, he's an expert, and he thinks that probably we need to check a defect called holocarboxylase synthase deficiency. See, I could remember that. I'm an engineer, but I still remember that holocarboxylase synthase deficiency. So basically, there's a defect on that particular genes, and he checked it, and he found that yes. And the way to fix is by prescribing her with biotin. Okay, now biotin is vitamin B. She's been taking that ever since, and now I think she already graduated with the bachelor degree in economics, right? Now, looking it into another scenarios, there's another boy who's also having the same disease, but he's not been diagnosed and not treated. He's barely able to calculate, and some lead to death. I mean, that is the kind of thing that, knowing it, hunting it down will actually save life at birth. So we have what we call it the neonatal ICU or NICU, that we have a window of opportunity, 48 hours. So there must be rapid sequencing facilities there to be able to get all the kids that seem to be having neurological disorder, check their genes first and then hunt them down, okay? So that actually explains the motivation of the project. So I'm not going to go through everything because I've got too many slides, and I have to be sure that I need to end this one before 10, and I have to have another meeting. So the vision and mission of this project, of course, we want to embrace the genomic medicine and making the genomic medicine become a part of a medicine in this country. That's for sure. Okay, and if you look at it, I used to actually have another slide talking about the generation of medicines. I mean, this is also interesting. What are the generation of medicine? The first generation is witchcraft. So you go to the voodoo and they just, try to do something. Luckily, you just, get cured by yourself. That's the first generation. The second generation is this generation when Chinese medicine, Indian medicine, they start learning about herbs, medicinal plants, and they start collecting their knowledge, accumulating, and they know what kind of thing that would be able to cure whatever thing that you are having. So that's the second generation. The third generation is the stat-based type of degeneration, which is the generation we are in now. Okay, so the doctor will go, will actually diagnose based on certain records that they have collected, like checking your CBC, like complete blood counts, looking at the profile of your biochem, so on and so forth, and they will just decide, probably you will have this. Or you're coming with a runny nose and having fever, probably you have a cold, catching a cold, or having a flu, something like that. But it's never precise. That is the medicine that we are having. And the medicine that we are having also include the kind of prescription that you have been given. Like I'm taking Tylenol, maybe 2 pills. Okay, some people said that I only take 1 pill, it's enough for me. So those are the variation. But when the drugs company, when they create drugs, they just test, they do the clinical trial. They do the trial and they say, okay, it's enough. We can market this drug already, right? They never actually pay attention to the small details that will actually cause some changes in terms of the quality of the treatment. So that is the third generation that we are in now. So the fourth generation is this generation, when you actually try to find the supporting information that can help you decide what type of the treatment you should be giving and what kind of the thing you should avoid that can give you prognosis that can predict your future. If you are born with the BRCA1/2 mutation, of course, you're not going to have the cancer just yet. But when you grow, the cells start divide even more at a certain stage. Of course, BRCA1/2 is the gene that actually fix errors, right, in your DNA. So if that gene defect is not going to cost you anything unless you start producing a lot more of the cells, a lot more of the defect DNAs. So if I can just tell you this profile, then of course, you know what you should do in the future. You could go like Angelina Jolie, or you could actually avoid certain things that would, you know, induce you to become cancer. So that is the kind of vision that we have. And I'm here, I'm right here. So to have the project done, you start noticing the challenges. Huge amount of data, I said, 120 gigabyte. So that is approximately your iPhone, 128 gig that you hold in your hand. And are you able to actually open the file using your notebook computer? Of course, but going through all the information inside is going to be a daunting task. So that's bioinformatics, right? You need to actually go and then try to extract a certain information out. Bioinformatics, I'm never going to say that bioinformatics is a tool to diagnose or anything. It's actually a tool to generate what we call it a hypothesis. But a good hypothesis, okay? Do not get me wrong. Do not go out from this room and say that, okay, I'm using bioinformatics. I'm going to use this bioinformatics to do a lot of things. And those things that you are doing simply generate new hypotheses, because later on, you're going to have to validate it. Bioinformatics can never be used as a validation tool. I mean, that's the thing. That's a fact, okay? So that's what we do. All right. So this one, okay, people would call it a population genomic project, because you're actually interrogating a large population and doing some study on it. So on one hand, we could diagnose all those volunteers that come into our project, you know, trying to figure it out what went wrong with them, or what kind of thing they should be worried, you know, because some of the things they never actually know that they have in them. None of us, including myself, are normal. Each and every one of you will carry pathogenic mutation, okay? But when you have a certain disease, you need to find out what is the etiologic mutation that caused your symptom, okay? But you already have pathogenic. It may not reveal itself to you unless there are certain conditions, you know, like happen. So yeah, so we do the population genomics, okay? We somehow seize an opportunities because Thailand is like a melting pot. If you look at the map, okay, our boundaries connect to Malaysia from the south to the east. We connect to Laos, Cambodia, and perhaps Vietnamese, of course, you know. And on the west, we have like Myanmar. In the north, we have China, Chinese people. So somehow, our genetics are the mix of a bit of everything. And I've got another researchers who are actually working on studying the population genetics of the Thai people. And we have seen some of the substructures that are similar to some of the people, you know, around here, and dated back even before our history. You know, history is defined as something that you recorded. So before you record it, then there's no history. But that actually happened. You know, who are Thai? Why are we here? I mean, all those questions that can be answered by looking at the data here. So so we're going to do the comparison of the DNA sequences in a population. That's mathematics, right? We're going to do identification of genetic variation. So collection of the genetic defect commonly found in this region. Would that be good? It is extremely good. Why? For example, if you are in the States, okay, I just need to compare with people from the States. You are not going to suffer a lot with the disease called thalassemia, which is the anemia. Okay, somehow your red cells Okay, if you have this defect, it's not going to be a good red cell because the proportion of the alpha and beta globins are not balanced. So somehow you cannot form a good red cell, right? That's thalassemia. Not going to happen much or very little in the States, but it happened a lot here in this region. So suppose we have found a mutation that we suspect that this patient is going to have, but we don't know if this is a good mutation that we should pursue. We cross-check with the database produced by the state, a collection of the defect or a pathogenic mutation in the state. We have found that, oh, this is so rare. This probably is the culprit, but actually it's not. It's so common among here. So it actually misled us to actually pursue in the area that we should not be pursuing. So if we have this collection, we know that somehow the frequencies of the mutation that we have collected here, this happen to be something that not so often, then we can pursue and then of course it save a lot of the cost because bioinformatics just only provide you with the hypothesis. You're going to have to validate it if it's so true, right? So that is the thing, and we have created the first database that we have collected from the exome sequencing. It's not a whole genome, but we just showed it. Okay, we have this one here, and we put it online so people can actually come and check it, even though it's not so complete. The complete one is coming, so the whole genome. Okay, we do the population certification and so on. Okay, so if you look at the National Genomic Project across the world, this is probably the latest one that I've shown. I'm not sure you can see it, but you will see that Thailand is having the active project now. Other countries also are starting, and we are talking, and WHO are also talking about including the genomic medicine and trying to actually find a way to help the people that are underrepresented, like for example in here, okay, and perhaps the blue ones are not well supported. I know that my friends in India, we know that he's working on population genomic, but the indigen, that's what they call it, started like a while and they just stopped. No more, sequencing or anything. They're just making use of that at the moment. So I guess this type of thing, is going to have to be a long-term collection and fostering, supporting the new medicines in the country. So we have, we are in the press here. We wrote an article that, okay, we announced we're going to do this project about a couple of years ago. I was the co-author of that paper too, I think. And here is the operational plan. I show you this setting because bioinformatics alone will not get you that far. We have actually worked seamlessly with other organizations as well. So we have about 29 so far medical center that help us to recruit individuals from all over Thailand. And when they recruited, they consent, the patient, the volunteer consent, and provide us with the blood. The blood will be shipped to another center called the Department of Medical Science, which is belong to Ministry of Public Health. We do DNA extraction. Only one place do the DNA extraction. Why? Because we want to have the same standard, so that the DNA, when they ship to the company will have no complaint, and the result that we get will actually be standardized. Okay, so that's what we do. Okay, the DNA will be sent to the largest sequencing center in Thailand, harboring 2 of the T7, which is the MGI-6 T7s, the largest in the market. NovaSeq 6000, that's also the large platform from Illumina. T7 is a large platform from BGI. So they actually have 2 of the T7. To tell you why they call it T7, because I think it generates the amount of the base is like 6 terabytes or 7 terabytes per run. Okay, so our project will have 48 individual being sequenced in 36 hours. 48 individual in 36 hours. So that being produced. So in about a week, we'll get about 2. Okay, hello. And then about a month, we'll get 1,000. A year, hopefully, we'll get 10,000 or more. So that is a kind of the thing that we're dealing with. So we have only one company doing that, so that the fast queue or the base call will actually be the same standard. Now, that's standardized. The variant calling, the base calling is when you sequence it. You call the base, ATGC, okay? The technology that used, they may be varied. But this technology will actually give you the base call that you will be using to do the variant call. It happened here, this place, okay? So we established some of the hardware that will help out with the project, because our concern is not just only get the data correctly, but also get data quickly. Because when they are doing the sequencing, they also need to collect money from us. But the way that they can collect money is when I say, yes, the data quality passed, so that's bioinformatics. So we check all the data quality metrics that has been sent to us, and we report it back to them so that the granting agency would be able to pay. We just paid the first batch, 494 individual to them, that cost about 5 million baht. Okay? So, and after this, we'll actually be sent it to the interpretation unit. And as I said, bioinformatics never do the task of interpretation for the medical doctor. We just generate a hypothesis so that the doctor will actually take a look at our hypotheses that we show to them, and they will make a decision based on their experiences. Okay, not us. Do not try to actually make this one automated so that it will just produce something that will be used in medicine. It's going to cause us a lot more problem. And this is my design, but sketch of the design. The cartoon that I showed it here actually exists. Some of the icon I use is actually exists in terms of the implementation that we did it here. Okay, we have here NASDA. Okay, we have the three stations before us, right? The medical center, the DNA sequencing, this one, and also here. Okay, and we have this as the last unit, right? We use Docker, okay? We use Kubernetes, okay, to actually oversee our hardware, okay? So that the Kubernetes will actually happen to help us with the management of the resources. We have, of course, the document database, you know, instead of the relational database, you know, right here, we do use it, okay? And we have, of course, the relational database implemented. So all these has to be done in concert in order to deliver what we call a service or hypothesis back to our partner, which is medical doctor. So I'm not sure about the other, but what I did here is that everything that gets sent to us will actually be pre-processed. The term pre-processing I use refer to, first, the quality checking. Second, the variant calling. We have to actually call the variant. We use, of course, the standard, like the broad GATK, that will help us do the calling. Hopefully, in the future, we'll have our own telomere to telomere reference. We are in the process of making that, but it will actually cost us extra because we need to have a long read sequencing done for Thai individuals. At the moment, I have about 60 of the long read sequence of Thai individuals, but the depth of those long reads probably are not enough, so we may need to actually do more in order to get that. So that's talking about the future, but it's a lot of work, you know. I myself will not be able to actually finish everything, okay? What else? Okay, variant calling. And then we go to annotation. We cross-check with the database like ClinVar, for example, some of the existing database that already has something annotated. So we annotate it back to our variant. We also have the effect predictor, functional effect predictors, like the DB and SFP. So that has to be implemented along the way. So everything that I'm talking has been pre-processed and put in our database. Now, the task next, I'm not sure, I think it might be in here, is a software that would get all the things that I put in a database, present it in a way that the medical doctors can change their hypotheses. See, I start having a family where parents are normal, but the kids are abnormal. Two hypotheses I can think of right away is first, the kids suffer from the autosomal recessive. So both parents are the carrier, pass on the defective gene to the offspring, and offspring is 1 out of 4 getting that defect. Okay, so if this is a hypothesis, then what you need to do is to use bioinformatics to actually gather all the mutations, all the variation that fit into this model. That is, both parents must have heterozygous and the offspring or the proband must have homozygous, right? So that's one hypothesis. How about another hypothesis? De novo mutation. So both parents are normal, but somehow the kid's so unlucky. So they carry the mutation by themselves, happen during the deontology at birth. Then you're gonna have to change, gathering all the mutation that, no mutation for the parents, but the mutation on the offspring. So that is a hypothesis generation that requires bioinformatics to actually go over everything that you have pre-annotated, pre-collected, and then bring it out for them. Now, another thing is that compound heterozygous. What is the compound heterozygous? So you have defect, but partially 2 positions in the same gene, but 1 position from the father, another position from the mother. So you end up having a defect on both of your DNAs. And this is very difficult to detect, because in short read, you will never know the phase of the DNA, because once you sequence, your DNA get blended, no phase, right? And this compound heterozygous is also a challenging one. I could try to actually make use of the tool to predict the phase for this certain region, but there's no guarantee. All the tools actually give you the probability, okay? Another thing that I have added into here is that I collect the stat. How often the 2 heterozygous happen to be on the same gene? I mean, that type of thing is a stat. So I put this one up so that the medical doctors can also cross-check with the stat that we have collected. And they will either be certain that this one could be, or they could actually reject this hypothesis. Because it's so common, like you found the 2 heterozygous together, so commonly happen in there. So these are also bioinformatics. See, a little bit of the detail that you have to pay attention into constructing the software for the user to use. You have to bring in, you have to know all those and bring them in. Okay. I forgot. You see, to actually organize all these stations to work in harmony, you're going to have to create a software for them. This may not be bioinformatics, like for example, the enrollment software. It's not bioinformatics, but it's an IT software that you have to create. The consent has to be e-consent. Everyone must actually show their intention in joining this one. Otherwise, you should exclude that. How do you know if each individual comes in and will not be repeating? Like for example, I check into the south because I have the problem with my eyes, and then I go to the north, I check again with the cancer problem. So ended up, I have 2 copies of DNA in my bank. So Yes, has to be done. Again, I step in and create the software, and they are actually using it. It may not be a great software, but we're making do because it has to be done. Okay, and how about collecting the phenotype? At first, I thought that, okay, we will just create something that will actually collect the phenotype for the medical doctors. And I found that it's a lot more difficult to do it. And there's a consortium that created a software called Phenotypes. I mean, you might be interested in also checking this out. They spin off and then become a company called Phenome Central. And that one's been used by the doctors, like rare disease and undiagnosed disease doctor. So we got the source code in Java from GitHub, and we install and then modify whatever it is that needs to be done and install it locally so that they can actually use this one. So this is very important because it will collect the human phenotype ontology, HPO terms and everything, and it can actually help construct a new hypothesis on what genes will actually be the one that they should be looking at. Because they can also come with their own hypotheses that, okay, maybe the defect on this gene, can I check if there's any mutation on the gene, right? So that phenotypes will actually help. And this one, for those of you who are actually familiar with LIMS, or Laboratory Information Management System, you will see that LIMS actually work in your own unit on every step of the way when you actually perform the experiment. But in this, it's actually end-to-end management. You see that 5 stations we have there, 4 stations need to be tracked, to see if there's a good flow of the data being populated from one to another station. So we also create another system we call specimen management system to oversee and to actually cross-check, like for example, DNA being sent to a sequencing lab, the DNA has to be in good quality, and both must agree on. So once you send the DNA to the sequencing lab will also check the DNA again to see if they would take that one and do the sequencing. Also, when they produce the data for us, we also need to check the quality, and we challenge them. So that will actually be the task of the specimen management system. And that is being used at the moment, okay? So the enrollment I talked about here, so, the phenotypes, yeah. So that is a link. It may be changed, okay? I will just, like put it out so that people can use it now because they're actually collecting the phenotype at the moment. SMS, yes. We have the end-to-end checking. We have the reporting system on each station. Okay, here. We talk about the annotation, variant mapping, right? So the DNA come from here, go to this machine. This machine is about 181 centimeter tall, so it's a bit taller than me, for this much, quite big. And the flow cell will be loaded right here. There's a small slot here, and they can load 4 flow cells. Each flow cell can sequence up to 15 individuals, but in order to make a good quality, they will do the sequencing for 12 individuals per one flow cell. So 4 flow cells will give us 48 individuals. So once you go in, there will be a robot inside, a small arm, getting the flow cell. They have the microfluidic inside. They will do whatever it is to sequence it. The light here, if it's green, it means that the flows has been loaded. So you break them into 4. So I've actually go in to check it out before. And when it's done, it will actually be unloaded on the side of the machine right here. Okay, this is a touch screen. It will actually show the status of each flow cell being sequenced right there. Okay, so yes, 2 of them. The data being sent to us. And if you are aware of the variant calling process that's done by GATK, GetK, you know that the first step is to align that to the reference. And that is a heavy task. I believe that if you don't do a population genomic project before, you would think that, oh, easy peasy, lemon squeezy, because you just do few. But when we become 100, become 1,000, that's a daunting task for us. And the part that actually kills us the most is the part that we combine everything. Before they call it combined GVCF, meaning that you need to know the landscape of the variation in your population. So you start putting them together just to see that. Okay, that takes a lot of time, and it cannot be parallelized. So that's a task that we feel that is very challenging for us, and it requires a design of HPC that is not a typical HPC that you use to do this molecular simulation. For the molecular simulation, when you do it, you will have as many nodes as possible, as many CPU as possible. You do not need much of the RAM because it can actually be done in massively parallel fashion. But when you deal with genomics, you need to have like large RAM, large scratch space, because Broad just introduced another way of doing the combined GVCF. They actually put everything into the database. And you know what? It demands us to actually have a large scratch space as large as 12 terabyte. So if you put SSD 12 terabyte, that is a lot to actually deal with this kind of thing. It's not so simple, okay? So it has to be designed in a way that will actually handle all these tasks that we have. Okay, I talked about the infrastructure. Oh, I talked about another challenge here. See, the DNA is stored about 140 something kilometers away from here. How do you actually transfer the data? You see, one individual is about like 100 G, right? So let's say that 50 individuals is 5 terabyte being transferred at one lot to here. So the network transferring 5 terabyte, that's huge. We stumbled upon this problem because at first we opened the link to transfer. We use a software called, I just forgot the name. The software is the layer 2 type of the network. It's just modified to expand the bandwidth of the network. So of course it can transfer quickly. But the problem is that all the services, the IP services, including like a WebEx, IP phone, get affected. So we have to actually move, the point of presence where the download happened, not here, but move it outside to another proxy, so that it will resolve that problem, and having the internal network transfer from that point of presence to this station. So everything is a problem that if you don't do it, you will never know. You never know. That has to be done. And right now, we are able to transfer 50 individual or 5 terabyte in just only less than 2.5 hours. So that's quick enough. Okay. So we set up the HPC here, and also where the sequencing lab is. The two system. One is actually act as the buffer, because if you keep on producing the data, you may overflow or flood the network. So you have actually put at least 1 petabyte over there just to accommodate this one. Okay, where and calling, I talked about that already. It's quite complicated the way that Broad has set up, but that's the best practices that we follow. So I think to make it as a standard, we have to actually follow with some standard that people are using. So that's what we do. The variant prioritization, yes, this is a software that we have created. There are so many demands from the medical doctor. They said that they want to see the BAM file, which is the aligned file, real time. So how do we actually get that? We integrate the IGV into our software so that people can actually zoom in, zoom out, and check if the reads are stacking properly. It's not overstacking, because if it's overstacking, it means that it aligned to some of the pseudogene. All those actually been implemented here so that they can see. We also provide them with the gene panel. Some of the panel has been accumulated by the Genomics England project. So that one we included in here, so that if the patient comes in, we check against the panel and present to them if they are interested in checking that one first before diving into discovery mode. The discovery mode is when none of the panel actually can give you the answer. So you suspect that it probably may have been the cause of the structural variant, the duplication, the CNV duplication or deletion. So that is what it is. Oh, we also published another work. You can also check the name PharmVIP because we see that the pharmacogenomics is the thing that people usually concern around here, and there's no standard tool. There's a PharmCat, PharmGKB that open as the public service, but of course, to process that, it requires some standard. So we adopt the standard here and implement as a tool, and this will be included in the pre-processing of the genome that's been sent to us as well. That's a database we have published since we started the project, even before we get the whole genome sequencing. We also work on the pathogen as well. We work with the division of the tuberculosis because we want to end the tuberculosis. Oh, wow. I just realized that I'm spending too much time on this. Okay, I will just try to wrap it up. And because I need to present another project to you. Okay, we also do the amplicon-based metagenomics. All these are the online tools that we have created so that it will foster the people who's doing research in here. All right, yeah. That's the end of the first project. But since I'm doing many projects, and this is only 2 out of many, so this also may give rise to some of the you who are actually interested in the machine learning, deep learning, or whatever learning that is. Okay, it's also a vital tool. For this one, we want to do just like we use the machine learning, okay, and the remote sensing, okay, to somehow predict the biomass, and we can also predict the carbon credit as well. The current carbon credit prediction require a lot of the effort from people to actually go into the area and then try to collect whatever data and somehow predict it, not the entire area, just only sampling some of those, okay? So we see that if we let it be like this, we will never be able to actually gain anything out of the carbon credit that we have here in this country, okay? We have to actually find a cheaper way to do it. And one way to do it is by using a remote sensing like a satellite image. But the problem or the pain point is that satellite image will not tell you how crowded the forest or the stem of the trees are, or the density of the woods that are underneath the canopy or the crown in the forest. You only see, bushes from the satellite image. But the hypothesis is this: the crown or the bush that you see from the sky, okay, represent patterns of the trees underneath, right? And of course, the color or the thing that reflect from the leaves of what you see from the sky can tell you what type of the trees you are looking at. Whether it's a dense type of the trees or it's a less dense type of tree. Okay. Everything you do, you have to have a hypothesis first. So if you have that hypothesis, how would you construct an experiment or devise a method to actually help you, either reject or accept this hypothesis? Okay, I hope that you keep that in mind. Okay? Everything in my life, even though I'm not a scientist, I'm an engineer, but I use this in everything in my life. Okay, I hypothesize first, and then I construct an experiment accordingly. So to do this, okay, we have a method like this, very broad method. We will first need to train the data, okay, using the field data, LiDAR, and hyperspectral. So my first hypothesis is that when I look at a bush, I should know how many trees underneath. So I must have a ground truth. So I send people into a plot, because there are a big plot here in Thailand called Mo Singto. Too bad, the time is too short. Otherwise, another trip that you should make is to go to our plot in Khao Yai, which is a nice place to visit. You get to actually do trailing up to the plot, and you see the gibbons. We know every gibbons in that area because we also tracking the route of the gibbons in that area. We know the sound, we know the breeds of the gibbons and everything. We know all the fruit, we know all the trees. In this 30 hectare, okay, quite large, right? We send people in. We measure the tree diameter, every one of them, about 100,000. Every 5 years, we do tree census. So we know the ground truth. We have a small plot as well, the blue one. So we know everything. We make use of this plot as a ground truth. We fly LiDAR over the entire big area. I'll show to you. Okay, so step one, high quality reference data generation of 64 square kilometers. So the ground truth, we send people to walk in, right? Now we use a plane flying over 64 square kilometers that encompass the plots that we have, just to get LiDAR or the three-dimensional of the forest. And then we first make sure that the ground truth align well with the LiDAR, and we upscale it to something that's larger, 64 square kilometers, and that become our model. The plan is to also find the type of the tree or the density of the wood. We are going to have another flight, okay, which is a hyperspectral sensor. Hyperspectral will actually fly over, shoot the sensor, and then receive the signal that bounce off the leaves. Hyperspectral will actually give you a profile of the frequencies that been bounced off, and it's been known that this signature can also determine the species that you're looking at. So that is what we are hoping to see. So we train this, and we create a machine learning. Okay, we create machine learning. So we have a row and columns, right? And then we have the output, right? We train it using random forest, which is a aggregated boosting, bootstrapping, and also the decision tree together. Right? So that gives us a model, which we use this one to predict, and we found that the accuracy is quite decent. We published 2 papers on this one, and we talked to the people, the company in Thailand, and they are interested in adopting this one as a standard to do the carbon credit prediction. We have actually made use of the height. We use the data from the GEDI that actually determine the height to make the model even more precise when it comes to prediction. So we upscaling, we publish the work, and we basically serve our hypothesis that the pattern you see here, actually refer to the trees behind the bush, and it actually fit the hypothesis that we see, and we hope that soon this will become something that people can use to predict the carbon credits. I think, yeah. So that actually will end my presentation in an hour, precisely. So hope it's not too long for you, not too bored. I know that the attention of the student usually go at the 30 minutes, and after that it's just going down the hill. But that's all I got, and I hope that I'm not giving you a bad presentation. And if you have any more questions, you can either ask in here or you can email me, and I would do my best to actually answer every question that you send to me. You can actually ask Travis or Amit or anyone too.
I didn't quite get, what kind of data do you give back to the people you're sequencing? Yeah, like, so if they have, you know, what form is the data and how much? I mean, if they have a mutation that it predisposes them to a disease, are you telling those people that, or what? Yeah.
Very good questions, because we've been talking among us, among the people who are actually working in this project. And the answer is that we are not planning to give everything back to the patient. The reason is this, very simple. Suppose I'm one of the volunteers. I got a cancer gene in me. The people who actually tell me this news will have to be trained. We call clinical geneticists. In Thailand, we only have about 20 or 30 clinical geneticists, which is not enough to actually handle this kind of task. So at first, we are going to do is we are going to get back the result of what they come to consult the medical doctors. So the report, even though I can print out everything, like the there's the ACMG, you know, American Medical College of Genomics and Genetics, right? A guideline on the predicting the incidental findings. I have that prepared, but I don't think the medical doctors will actually take that and give it back to the patient at this moment. Because if you are nervous, you could do something harmful to your life. You could just commit suicide or something if you don't get a proper consultation from the clinical geneticist. So that is what they think they will do at this moment. But we, at the same time, we're trying to actually build more of the clinical geneticists so that the genomic medicine will actually become a part of the public health in the future. But that's a good point. We are not giving everything, even though we can, but we are not. Good questions. Yes?
So do the physicians in Thailand get training on genomics? Like, how are you?
That is a good question too. You see, we're not ready. I think many countries in the world, including in the US, I think they're not ready. But they actually need to give more training to the people. Like when COVID started, you will start seeing that the US get hit really hard because modernization of the infrastructures is not happening everywhere. It just only happened in a big city, right? And that's what happened in Thailand too. Not everyone here in Thailand knows genetics. The medical doctors may not also know genetics so well. They may have been trained, but that's a long time ago. They may have forgotten already. You know, if I talk to some of the medical doctors, I may know a lot more in genetics than the medical doctors. Yeah.
Thank you, Sissades, for that wonderful presentation. Just continuing on from Amit's question, do you think there's value in coming up with software, genomic software that simplify the whole process of doing, you know, the variant calling and all that stuff, variant prioritization, even coming up with the report, so that the clinicians themselves could be part of that process, could be involved, could actually start maybe uploading the files themselves, looking at it and interpreting, rather than relying a third party to do all that. And I know it's not really ideal. I don't think it's perfect because they wouldn't know all the details. And when you simplify the process, there are some risks involved. But maybe it's a step, you know, forward to bringing them in that direction.
Yes, that's what we are doing at the moment. We create the software called variant prioritization. Actually, I changed the name. It become VAPP because when I put the term interpretation in the middle, variant interpretation and prioritization, the medical doctor said, no, you're not doing my task. My task is to interpret. You are not interpreting. So I changed that to VAPP, variant annotation, just like what Sip said, and prioritization, because the prioritization part is the part when they can come in and then generate their own hypotheses and then find out if whatever it is that fit to their hypotheses. So yes, we are having that. And we hope that this one will actually be something that people can access, because in other country, this software is commercialized, meaning that you have to pay a large amount of money, even though you don't use that every day, to subscribe that software, like Golden Helix, Congenica Serpentia, the foundation. You know, there are many software out there. But what I'm doing here is the first time that I actually doing it for the entire country, and hope that in the future we can actually offer that as a cheaper service to do. But if we don't start it, we will never know. And you know what, as if it's precise moment when we decide, okay, either buy it or subscribe it or do it ourselves. And I took another option, do it ourselves. It was quite risky, yes, because there's a lot more than what we think we have to do. You see, the detail that I described, those are just only small contribution that I mentioned to you all. But there are a lot more that we have to do in order to make the software complete, just like what Asif said, to make it easier for them to use. It's a lot harder, but yes, it's worthwhile.
Could you share what has the response been from clinicians in adopting? How easy is this to be used by them?
It's just beginning. I don't know. I haven't gotten feedback yet. I'm about to actually give a lecture on this evening. They will actually be able to give me some feedback on how easy it is to use, how easy to adopt into their normal process. That I don't know, because it's new. But I think that whatever we offer may not be as good as a commercial software, but it's better than nothing. So that's what bioinformatics is all about. We are learning here, and the student asks, if we can apply some of the things that we know into something that would help save lives, right? That's what Amit put that into the perspective. And this is what we are doing here. And I think the reason that Amit asked me to share with you, because he wants us to hear and see if one day when you are old enough, you would be like me.
Thank you, Sissades. It's really good that you presented this, because we are doing something similar, and I would love to learn your experience on this. Thank you.
And I have to apologize, Amit, because he's my boss, and I was answering to his call because I was busy doing this.
On quick last questions, do you have any course for genomic counseling started here? Because that's
yes,
we do.
We do. It's been opening for 1 year already. We need to increase the number of the trainees, and we also increase number of courses, including the bioinformatics like this. May not be hardcore bioinformatics like you guys. We need to actually get people, practitioners, to understand what they are about to use. Doesn't need to actually know how to program. Doesn't need to but understand some of the terminologies that they would see and, oh, okay, I got an idea. That also needs to be done, to make it successful. So the lesson learned, I guess, we are in a process of growing. And this evening, there's one company, I will not name that company, but basically they are going to open the genomic service, and they are interested in getting the help from us. And we are going to get together and then discuss about the possibilities. Yesterday, I wasn't here. I also went to the medical, the sequencing lab in the Eastern Seaboard. They also asked if they can also use this service instead of the sending the data to the headquarters. Because again, huge data, big data. One thing that I haven't actually shared with you is that there's an agreement among the scientists, that when you deal with this size of the data, we will never do like moving the data around. We will move the algorithm to the data. Okay, that's how people agree upon. Okay, the data will not be moving around. The algorithm would move to the data. You would come here and then process, something like that.
If there are no more questions, you can thank Sissades for the wonderful presentation.